I am thrilled to announce our first keynote speaker of West 2024, Dr. Irena Creed. Dr. Irena Creed holds a Master in Science in Botany and Environmental Sciences, as well as a PhD in Geography from the University of Toronto. Dr. Creed is the Vice Principal of Research and Innovation and the Professor at the Department of Physical and Environmental Sciences at the University of Toronto, as well as an adjunct professor at Western University in Ontario, Canada, where she served as the Canada Research Chair in Watershed Sciences for 10 years. She has gained an international reputation for creatively combining theory and practice to improve understanding of the links between hydrological and biogeochemical processes and their ecological consequences in freshwaters, particularly for at-risk human settlements. Today's talk is entitled Climate-Driven Changes in Terrestrial-Aquatic Linkages Contribute to the Rise of Cyanobacteria in Some but Not All Lakes. We encourage attendees to ask questions throughout the talk. We ask that you do this through the Q&A rather than the chat. If you submit questions through Q&A, we will be able to address as many of them as we can at the end of Irena's talk. And if you have issues accessing the Q&A, we know that some people may not be able to see it, but if you press the 3 buttons titled More on your Zoom keybar, you'll be able to access the Q&A there, hopefully. So with that, Irena, I will let you begin to screen share and take it away.
There we go. Well, good morning, everybody. It is such an honor and privilege to be here today. I am going to speak to you about something that I'm quite passionate about, but first, I too would like to give a land acknowledgement. I acknowledge the land that I work on at the University of Toronto. It is the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. And today it is a vibrant land that is home to many Indigenous people from across Turtle Island. I want to talk to you about something that is considered a global crisis. And in this 2022 World Economic Forum report, there was an opinion piece offered of why we need to fix the world's freshwater algae problem. From that report, I highlighted some text to explain why this is such an important issue. They call them harmful algal blooms, or HABs, and they have serious health, environmental, and economic effects on people worldwide. Just reading one portion of it, avoiding drinking contaminated water cannot completely protect the public's health, as contaminated lakes still threaten people, animals, and the environment. Part of that is because not only can you be exposed to it by drinking the water, but also by breathing in aerosols that are emitted from the water, as well as the food that is in the water. These, after being in contact with affected water, the short-term effects may include skin irritation, nausea, and even paralysis. And the longer-term effects are still being researched, but are already known to include liver cancer, the neurological diseases such as Lou Gehrig's disease, ALS, and Alzheimer's disease. So this is a problem that I'll explain later in my talk about how I first started researching it, but something that I'm very concerned about. So I wanted to introduce the freshwater algae of concern, cyanobacteria. And this is the one that many of you may recognize, the photo of Lake Erie and the western part of Lake Erie being known for notorious freshwater harmful algal blooms dominated by cyanobacteria. And so these cyanobacteria are on the rise globally and creating an escalating concern. Now, what exactly are cyanobacteria? They are prokaryotes that obtain their energy through photosynthesis. They have the anatomy of a bacteria and the physiology of an algae. They can appear in many different forms, single cell, colonial, and filamentous forms. They can form blooms and they can produce toxins. In the image on the left, toxic algae coming soon to a lake near you. It is incredible the prevalence of these harmful algal blooms within both large and small systems throughout North America, as well as other parts of the world. Lake Erie in the 1960s was declared dead because of these monster algal blooms. And some of you may recall some of the giants in water science in Canada, such as Dave Schindler. Dave Schindler was working at the Experimental Lakes area, and he worked on what was causing some of these major blooms that were killing lakes. And he discovered that it was phosphorus in detergents, and that because people would wash their clothes, wash their dishes, the phosphorus would go into the water and create these blooms. And it is his seminal work that led to the removal of phosphorus from detergents. And at one point, we thought that Lake Erie was resurrected, it came back to life. However, in recent years, as shown on the image on the right, you will see that these monster algal blooms are coming back. And the question is why? I also want to show you on the left-hand side that these cyanobacteria are no longer restricted just to the western basin of Lake Erie, but go through all 5 of the Great Lakes. One of the things that really drew my attention was in 2014, when we had a major episode in the western basin of Lake Erie, where Toledo, Ohio residents were warned that they couldn't drink their water because of this algal bloom. This had a microcystis bloom, and the microcystin toxin you can't boil the water and have the toxin disappear. And as a result, people had to start buying bottled water during this period of time. And I just want to take a moment and pause. Like, here we're looking at 18% of the world's freshwater supply, one of the greatest resources that we have in North America, and yet people could not drink the water because of the toxins living around Lake Erie and had to use bottled water. But this is where my research really started coming into this area. I was sitting at my desk, and at the time I was at Western University, and I picked up the phone and received a call from people working around Dixon Lake. Dixon Lake is a really important lake for Indigenous communities, as well as tourists who come there to recreate. It is based in Algonquin Park. And the person that called had this big concern, and you can see in the lower images why they were concerned. Dixon Lake and some of the other nearby lakes were closed for travel and camping because of an algal bloom. And so you can imagine in this if you look at the top right image, the remote sensing image, you could see a little lime green dot, and that is the lake that had this algal bloom. That was quite a concern. People never saw it before. They were wondering why was it there then, and also what they could do about it. So since that time, when again, that was around 2014, and people have been, the provincial governments have been compiling citizen reports, using citizen science to report when people see these blooms. And recently, a colleague of mine, Liz Favreau, compiled even more recent data on these citizen reports. On the top part of the plot, you'll see the number of confirmed cyanobacteria reports. So basically, someone calls in a report, and then the province will go out and test to make sure it is cyanobacteria. And on the bottom one are the number of water bodies reporting cyanobacteria blooms for the first time. And so what you can see is that the number of reports that have been confirmed are escalating at a remarkable rate. And also the number of new lakes being added are also escalating. Now, there's a cautionary note when you look at data like this. We don't know if public is becoming sensitized to these blooms, and so maybe they were always there and people just didn't report them. So sometimes you have to use other ways of science to say, are these real? Is this actually increasing? Or are we just observing something that people hadn't been observing before? That is something that I also looked at in my research. What Liz Favreau and her colleagues also showed is that when these reports were happening, they found that if you look at the left-hand side in 1998, that it was typically the peak blooms happened in the late summer, maybe August, September, October. But what they're finding is that the length of the bloom is actually happening over a longer period of time. It's happening right after snow melt in the spring and can go even far into October, November, and even December in some cases. So they're now not only being restricted to a certain window of time within the season, but happening throughout the snow-free season in these systems. So I'm seeking to develop a process-based understanding of the rise of these cyanobacteria in these northern lakes. And I consider many of the communities dependent on these lakes at risk because not many don't have drinking water treatment supplies. They depend on these lakes for their water as well as their food. And these are things that really concern me, and I really wanted to contribute to mitigating the risk of these blooms in these waters. So first of all, I want to take it right down to a single cell. And what do cyanobacteria need in order to grow? Well, we know that they need phosphorus and nitrogen. These are macronutrients, and the algae need them to grow. But they also need iron to grow. And so I'm going to go through what these macronutrients, what evidence is there of that changing, and what new ideas are needed to explain the cyanobacteria blooms in the far north. So as I mentioned, Dave Schindler, this is an iconic image of his experiments that led to our understanding of the important role of phosphorus loads to the risk of algal blooms. He had, in this experimental lake, drew a curtain throughout intersected the lake. On one side, he added phosphorus. On the other side, he did not. And what he found was he was able to create this algal bloom. So we know, and the current paradigm is that larger loads of to lakes of phosphorus will increase the risk of algal blooms, and phosphorus control, such as the one in the detergents, as well as now some in many of the water treatment facilities, has been successful in lowering algal biomass, and this must continue. But as John Downing published in just around the turn of the millennium, he said that whereas total phosphorus and nitrogen are important, cyanobacteria blooms can still occur at any total phosphorus, at any total nitrogen, and at any ratio of nitrogen to phosphorus. He said, but although the risk increases as phosphorus and nitrogen increases. So this was a concern because there's a lot of variability, and just focusing only on phosphorus was not going to be enough. So this made me ask the question, is it time for a new paradigm? Are there other causal factors linked to the increasing productivity of cyanobacteria? So we look at this and we know phosphorus can be directly taken up in the cell wall. With regard to nitrogen, which has now been an increasingly received a lot of attention because people feel that maybe it's not phosphorus. Phosphorus might be for green algae, but nitrogen might be for cyanobacteria. But when you look at nitrogen uptake, it depends on iron. And iron is also needed for other cell reproduction processes. So I ask the question, is iron the missing factor driving the rise of cyanobacteria prevalence? So let me tell you a little bit about iron. There's two forms that we'll be talking about today. The first one is ferrous iron. It's the Fe2+. Its dominant form is the dominant form in low oxygen waters or anoxic waters, and it's highly soluble. And what that means is that cyanobacteria can take it up directly. The other form is called ferric iron, Fe3+. It is the form of iron that is dominant in oxygenated waters, but it's not highly soluble. What that means is that it precipitates out relatively easily. However, if you do have ferric iron, even for short periods of time, it's accessible to all phytoplankton. But cyanobacteria have an iron scavenging system that allows it to outcompete other phytoplankton. So where does the iron come from if this paradigm is going to work, we need to understand where this limiting nutrient may come from. And so this is my simplistic cartoon of a lake. And as you can see, the three different colors at the top is the epilimnion. That's where you get warmer, lighter water. You then get the metalimnion or thermocline, which is the transition zone. And then below that, you have the hypolimnion, which has cool, heavier water. So the first question is, do cyanobacteria access the iron from lake sediments? And there's a lot of really interesting work done by many of my collaborators and colleagues in this area. You look at a lake, if the hypolimnion, that lower part, the sediments become anoxic, you can get iron 2+ from the sediments going up into the hypolimnion. And then from the epilimnion, you can have cyanobacteria who migrate up and down like going on an elevator. And when they need the iron, they can go down towards the hypolimnion, get access to the iron they need, and then move up to photosynthesize and reproduce. So this is one of the ideas of where that iron is coming from. But in the lakes that I looked at, the hypolimnion remained aerobic, oxygenated. And so only a few of them, which were really deep, had anoxic conditions. So this supply of iron did not seem to be practical in the systems I was looking at. So where else can you get iron? Well, another supply is dissolved organic matter, and that's what I call DOM, and the DOM iron complex. And this is where the cyanobacteria can access the iron through this iron scavenging strategy. And this work was first mentioned by a graduate student, a PhD student at UBC, who published in Science, and he discovered these siderophores in oceans. And so we knew that there was this ability of phytoplankton to use siderophores to acquire iron in really low iron environments. And so we wanted to look at this in freshwater systems. And so these siderophores, there's two different types, and I'm not going to go crazy on the chemistry with you, but one type is where either the cyanobacteria or bacteria or fungi can release the hydroxamate siderophores into the environment. They roam around in the water, and then they can deliver the iron to the cyanobacteria cell. The other type of siderophore is called the catecholate, and it is embedded in the cell wall of the cyanobacteria. And in this case, the cyanobacteria cells move around, and when it finds an iron 3+, it can lock it in. It's like a lock and key mechanism, and then transform it across the cell wall. But here then the question is, what controls DOM iron flux to that epilimnion? And here's where I really get into the area of climate change and terrestrial and aquatic linkages. As in the introductory comments, someone mentioned that I was a Canada Research Chair in Watershed Science. I spent most of my career working in the catchments, and this work really brought me to the endpoint of what a lot of those catchment processes are to the lakes and to cyanobacteria. And my thinking was that it's the watersheds or the catchments that provide the sources of iron that can fuel the cyanobacteria. Now let's look again at this landscape with Dixon Lake and that lime green, it almost looks like a kidney-shaped lake. And you look all around that, and all the red color are sugar maple forests, the green are conifers. And the question becomes, why that lake? Why just that lake? It's not many lakes, it's just that one lake. What's special about that lake? And so this is where we have to go underneath the canopy. Some of the work that I've done a couple decades ago now is something called cryptic wetlands. And on the left, you can see the sugar maple forest and the leaves are turning color. But below, you see these small features on the land. You have the upland on the left, and then you have a lowland, and then these inundated, saturated soils, the outer wetland and the inner wetland. These are what I called cryptic wetlands. They're cryptic because they're hidden from our eyes because of the canopy that covers them. And some of the research I ended up going around and using the very high technology of rubber boots. And you walk around the watersheds and you look for where you get the squishy part where the water comes to the surface, and mapped out these cryptic wetlands. And now, by the way, there's much more sophisticated technology using LiDAR, airborne, and satellite imagery to map these out. But in this first paper, it was done by the squishy boot method. And what I found is these many catchments had different proportions of cryptic wetlands. Some went up to about 20%, whereas some, like that's in C38, and then down below in C33, less than 1%. And what I found was that ones that had a lot of cryptic wetlands, they exported a lot of carbon and carbon plus nutrients, including nitrogen, phosphorus, and iron. So when we look at these catchments, we also know that climate change and climatic variability is occurring. And I've published work on the fact that it's causing what I'm calling an intensification of the hydrologic cycle. And other people have reported on this intensification of the hydrologic cycle. The way that I've presented it in my papers is basically dry periods become drier and wet periods become wetter. And so when you look at the left-hand side, very close to Dixon Lake, you'll see that overall the temperatures were rising and the precipitation falling and the runoff falling. So overall, this landscape is becoming warmer and drier. However, when you look at it on a month-by-month basis, which is the figure on the right, and you look at the potential evapotranspiration over precipitation, which is the dryness index, if you get a lower PET over P, meaning if the lines go down, you get more water on the landscape, and if the lines go up, you get a higher PET over P, you become drier. And what we found is that during most of the year, it's becoming wetter from January to April and from September on. But in the summer, you're getting really dry periods. So in this case, the manifestation of the intensification of the water cycle is that the summer months, dry periods are becoming drier, and the shoulder seasons are becoming wetter. So what implication does that have for DOM, dissolved organic matter? Well, then I went to two catchments, one that had like less than 1% wetland. That was the one on the left, catchment 35. And then the one that had 20% cryptic wetlands, that's the one on the right, catchment 35. And I looked at the long-term trends in dissolved organic carbon export. The one without a wetland, you could see that it declined. It just continued to decline. And so in this way, climate change is deactivating the DOM export. But on the right-hand side, the one with the wetland, you see the trend going up and up. And in this case, climate change is activating the DOM export. And these two catchments are side by side, literally. I can walk across the forest harvest road to access the other one. So two right next to each other, and yet they respond completely different because of these cryptic wetlands. The other thing I want to show you on the bottom is, again, the January to December, the 20-year time period trends for each month, that in the September, October, November, you get huge declines. It's really concentrated there. And yet in the one with the wetland on the right-hand side, you can see a big major pulse happening in October. And remember what I said, that some of the cyanobacteria blooms are extending into October, November, December, and maybe they're getting a fresh supply of the dissolved organic matter that's feeding them, enabling them to be successful. So based on that evidence, I propose that terrestrially produced DOM iron is fueling cyanobacteria in these northern lakes. So then I wanted to look at the direct correlation between dissolved organic matter and cyanobacteria density. So what do you see? Is DOM related to an increase in cyanobacteria? Well, nope. There's no correlation happening there. And so the high density cyanobacteria lakes you could see happen on the left-hand side in the blue dots. We don't seem to be having any regulation or influence by dissolved organic matter. So I didn't want to give up there. I started thinking, okay, is DOM a sink or a source of iron for cyanobacteria? And to do this, I looked at DOM composition. And there's something for those of you who understand humic substances, which are relatively refractory organic matter. I looked at the humification index and thought, well, maybe that might be a factor that would explain what I'm seeing. And so is DOM a sink or source for iron? It depends. When you look at very high humification index, where it's really refractory, very aromatic, you're finding that there's no response with cyanobacteria. The cyanobacteria seem to increase where there's a lower humification index. So this makes you think that more labile DOM might be linked to more cyanobacteria. And so I then went into the chemistry a bit more with the iron. On the top, you see iron hydroxide. It has a very weak iron binding, and eukaryotes can access it. In the middle, you have labile DOM, which has moderate binding, and that's where cyanobacteria can access it. That's the hypothesis. And then you can get really refractory DOM. That's the one with the higher humification index. And that has a very strong iron binding. And the idea here is that neither eukaryotes or prokaryotes can access it. And so this is when we started to see some evidence that siderophores, which are basically some of the more labile DOM, you see an increase of cyanobacteria density as these siderophores increase, and those siderophores can access the labile DOM. Pretty cool stuff. So we do find evidence from what was first mentioned in the oceans and now the freshwater systems. And now I've given you evidence that it's really kind of catchment specific. It depends on the processes within individual catchments. And now I want to see, can we explain why some lakes bloom and others do not? This is where I now go from the ground in the squishy boot method up to satellites and look at what can I see from a satellite and cover not just one or two catchments and one or two lakes, but to go to tens of thousands of lakes. And so I used what was freely available, Landsat imagery. We have it now since 1984 to present. It has about a 2-week repeat cycle, 30-meter spatial resolution. Did I say it was free? This is one of the coolest things because this incredible resource is freely downloadable by everybody. And so what I did was I created a 30-year time series of algal biomass to explore the spatial and temporal patterns of algal blooms. Now, this is a fancy two-way ANOVA. Some people will do it in a table, and I did it in a cartoon. But basically, you'll see that on the top left are my landscapes with Lake 1, 2, 3, the images that go from 1984. In this case, I went up to 2011. And I measured the chlorophyll by using the satellite imagery. I was then able to break down into what variability could be explained by time, what could be explained by spatial patterns, and what were the interaction between space and time. So when we look at the top right, I could say that 20% of the variability that I observed was variation due to regional temporal controls. And 36% was due to variation of the intrinsic properties of the landscape. And there was the majority, which is problematic because it's really tough to be able to disentangle the space-time interactions. But the majority, 44%, was variation due to lake-specific factors. And so then I went further to see what more I could learn from this. First of all, I just want to show you the 30-year average of the peak algal biomass that was observed in this landscape, which is basically the northern temperate forest area through the Algoma Highlands and the Madawaska Highlands. Some of you who may be familiar with this area, there's Sault Ste. Marie, Sudbury, and it goes into Huntsville and Bracebridge. And what I want you to do when you look at this image is to see, I mean, it's a pretty image, but it also shows remarkable heterogeneity. Like I showed you with Dixon Lake, one lake has a high value, the lake next to it does not. As I showed you with catchment 35 and 38, the catchment specific processes vary. And sure enough, what you see here is that some lakes in this landscape, which does not have a lot of human influence, have hyper-eutrophic lakes, and some of them are oligotrophic, if not hyper-oligotrophic. And so then I wanted to say, how can we predict whether one lake is going to be likely to have a bloom and another one not? So this is where we start thinking about the landscape and the functions that the different catchment properties deliver to the lake. And so I looked at catchments and their nutrient export potential. I looked at ecotones, that transitional between the catchment and the lake, including the wetlands and the littoral zone. And then I looked at the lake itself in terms of what was the probability of having nutrients resuspended from the sediments versus ones that are only coming from the catchment. And then on top of that, I also looked at air temperature and precipitation. So one of the things have built a reputation for is looking at catchment topography and understanding how we can come up with GIS and remote sensing tools to look at the functions of catchments because of their topographic features and make predictions about nutrient loading and consequences within these landscapes. And now I'm going to take you through this carefully, but I'm really excited about this finding. And the reason is because as I showed you with all of these simple catchment metrics that I was able to extract from digital elevation models, I was able to predict a lot of the variability in the lakes with the algae. So I'm going to take you to the top. And the first most important factor was lake volume. If you had smaller lakes, they were typically, and warmer lakes, they had the highest algal concentration. They were the most eutrophic. And so many of you, if any of you follow this literature, you might be familiar with Hans Perl work, and he's an amazing American scientist. And he published a paper like Blooms Like It Hot. And the idea was that blooms like it hot if, in this case, they are small and they're able to get warmer. That's where you get the bloom. So now let's look at the other part of the lake volume, the larger lakes. They don't like it. They don't necessarily like it hot. There's something else driving it. And so if you go down the tree from the larger, you'll see the dynamic ratio. The next thing that the lakes really responded to was the higher resuspension from the lake bottom. That's where you get phosphorus and perhaps iron that is locked in the sediments that get resuspended. And so that was the second explanation for what would drive algal blooms. As you continue down the left, you then see that you get higher loading from the littoral zone. And if you go to the tree all the way to the left, you'll see that those are the ones that never really turned. They were consistently oligotrophic. And so this was a way that you could take a landscape that is not often accessed by humans, and yet use a combination of theory and the practice and applying state-of-the-art technology to be able to make predictions of why some had certain algal levels and other values for tens of thousands of lakes. I also wanted to show, and this was something I did not expect at all, I thought climate change, everything's getting warmer, that all the lakes would soon, gradually show signs of becoming more and more eutrophic. And what we found was that only 4%, 3.6% of the lakes showed a trend of an increase in chlorophyll. And 7% of the lakes, it was the other way around. It was an oligotrophication. which is something I wasn't expecting. And of course, now we have a whole new research that we can follow, is trying to understand why some lakes are becoming algal enriched and why others are becoming algal poor. This led me to think, and by the way, just going back here, of the total, only about 10% had a trend. And so, we're seeing these changes, we're feeling these changes. We go to the communities and they say that they're seeing some evidence of change, but our technology is not able to predict it. All we're able to predict is a total of 10% and 3% going up and 7% going down. But this is where we want to talk about early warning systems. And some of you who know early warning systems from an engineering perspective, I'm going to present one of early warning from an ecological perspective. And this is what we talk about, a gain or loss of resilience of lakes that might lead to a regime shift. And by here, we mean a regime shift from a system being oligotrophic, all the way going to unstable and becoming eutrophic, or the other way around, as I just showed you, that there's evidence that it could go either way. So I did a series of statistical treatments of the data, and this has been submitted to ecosystems. It's still under review, but the methodology has been published in earlier papers when I was looking at changes in stream chemistry, lake chemistry. And so what we do is to take the time series, and you want to basically remove any trends from the time series. And then you want to remove any oscillations from the time series. Some of you may be aware of climatic oscillations, like the Pacific Decadal Oscillation or the El Nino oscillation. And we didn't want things that were stationary. We wanted to, or non-stationary. We wanted to look at what was left over, what we call the residuals. And so we then removed the trends, removed the cycles, looked at the residuals, and wanted to see if there was a pattern in the residuals. So on the right-hand side, we see that if there was a pattern, if there was no pattern, it would either be stable eutrophic or stable oligotrophic. And so what we found here, again, these are the residuals. And you see some are stable on the right-hand side. You see ones that are really going all over the place, really unstable lakes. The standard deviation below that, the chlorophyll below that, and you can see that I found that in that 10,000 data set, 10,000 lake data set, about 1% were stable eutrophic, about 33% were stable oligotrophic, and about 20% were really unstable. We then went to the right-hand side and we wanted to see, was there a pattern in the residuals where it was decreasing over time or whether it was increasing over time? And what, if it was decreasing or increasing, was it shifting towards more eutrophic or less eutrophic? And this is what we found here. First of all, on the left-hand side, destabilizing from oligotrophic towards eutrophic, we found 33% were shifting, 16% transitioning, 17.9% unstable, and then we found 7.8% transitioning towards that new state. And similarly, same discussion on the right-hand side, but the other direction. How many were going from eutrophic to oligotrophic? And you can see that 19% were shifting towards unstable to an oligotrophic system. So these are examples of early warning systems that you can look at in the residual of your data. I just want to show it to you here. What did the residuals look like? The destabilizing ones on the top, you can see the residuals increasing as you go from 1985 to 2010. And the ones that were stabilizing, the residuals were decreasing over the time from 1985 to 2010. And again, wanting just to show you a map about what the distribution of that looked like. The loss of resilience was observed in the subset of lakes that I had a lot of the GIS and remote sensing data for. No real spatial pattern in terms of the region, but now we know that we have to look at individual catchments and their basins to be able to predict if a lake is going to be not only changing, but early warning systems of that change. And so what did we do? We ran a random forest, which is like the regression tree that I showed you earlier. This is one that's run 100 times. And so it's called a random forest. And the number one factor that was important in predicting destabilizing lakes was the cryptic wetlands, the wetland cover underneath that forest canopy, followed by lake volume, the littoral zone, and then the climate change factors. So I'm just going to conclude with this kind of key message. These changes in the atmosphere and the terrestrial systems can result in disruptive forces to aquatic systems. These changes are often slow, and the evidence I showed you today, where only 3.6% showing one direction change and about 7% the other, not many of them were changing. But they are becoming fast as a result of anthropogenic climate change. And some of them might be approaching these tipping points towards fast changes. And so when I showed you the gain and loss of the residual patterns, the resilience as indicated by the residuals, that's evidence that you might be reaching a tipping point and shifting from one regime type to another. And obviously, these pose risks to the northern communities that often don't have the privilege of being able to have water treatment facilities nearby. And I just wanted to acknowledge the incredible graduate students I've had the privilege of working with. And this work really reflects all of their efforts. And that's the end of my presentation. Thank you.
Awesome. Thank you so much, Irena, for your talk. It was very insightful. It was interesting to see this connection between human impact and climate change and ecological consequences. And I also really enjoyed seeing the transition from your boots on the white ground to the Landsat data as your career went on.